you very much, Anne, for these kind words, and thank you very much to the Presidium of this university and to Anne and colleagues for having me here today. It is a great pleasure to contribute to the Global Science Challenges Week in Grenoble. And it is a shame for me I cannot stay any longer than just this morning because of other commitments. But coming here yesterday and seeing the beautiful scenery of Grenoble and the Alpes, that was already a big reward for me, and being here is another one. Yeah, I'm not a neurobiologist. I say it from the start. I am an enzymologist interested in enzymes that play an important role in neurobiology. And this is why I'm touching the field of neurobiology and why I'm contributing to this field in collaboration with other colleagues. So this is what we all learn, or at least the older generation may have learned when they came to school, and we were not very much tempted to do our housework at home. They always told us in German it is, was Hänschen nicht lernt, lernt Hans nimmermehr. So that means a tree must be bent while it is young. Why is that so? It is, in fact, the case if you look at the plasticity of our brain. And this is what we see. I can use this. The brain's ability to change in response of experiences is going down rapidly, as you can see here. In the age of 10 years after birth, we have lost more than half of that capability. You can compensate this amount of effort, you can compensate for this decline by efforts, by mental and motoric efforts, so that we have this orange curve here that can compensate, but you see with age you have to invest an not extreme, but a significant amount of your time to compensate for your neural decline. Physical and mental activity attenuate the cognitive decline. This is an important point, and I wanted you to remember this for the rest of my talk. This decline concerns different aspects of neuronal plasticity. The increase in neurogenesis is going down with age. There is an increase, the increase of, or the motoric and mental activity increase the neural plasticity, neurogenesis, and also synaptic strength. And this has to do with cognitive functions. And all these processes, and this is why I'm here today, have to do with glycans. And now I do a little excursus into the material, into the domain where I am more familiar with, and this is glycobiology. But before I do so, I show you the structure. This is essential in learning and memory processes. And this structure is called the hippocampus, and it is the green structure that you can see here, or excuse me, or here in the schematic scheme, it is the red structure, and this is the anatomy, the morphological anatomy of this structure in human. Because we do not much studies in human, as you can appreciate, we do most of our work in mice. And this is why I show you also the hippocampal formation in mouse. And this is in the computer tomography, and this is how it looks in the histology, or in the scheme of the histology. And I'm mostly focusing on this type of structure, where you have the dentate gyrus in this area. And the dentate gyrus, there is cells that I'm addressing a lot today. And those cells are sending axons into this area here, which is called the mossy fibers or the Schaffer collaterals. but this is not what you need to remember. But this area is extremely important in terms of neurogenesis, lifelong. As you can see from this slide, this is a very recent publication in Cell Stem Cells, a very nice one. Whoever is interested in this field should have a look to that publication. It's really nice. And that study demonstrates that in this area, at the bottom of this area, this here, is a subgranular layer. And in this subgranular layer of the dentate gyrus, we have lifelong quiescent neuronal precursors that, based on a stimulus, can start a asymmetric division and then produce this green guys here, and those intermediate neural progenitors then go through different stages of differentiation and can produce new mature neurons that are integrated, and this is very important in the brain, correctly integrated into the brain in the hippocampal formation. And here comes the structure that I like the most, and this is the sugar polysialic acid, and what polysialic acid is, I tell you in a minute. And as you can appreciate from this slide, all this process of differentiation, but no more the mature granule cells, are polysia positive. Polysialic acid is a marker for this differentiation, and of course not only a marker, but it has a function in this differentiation process. So here we are back to glycobiology. Each living cell, and this is a standing rule without any exception, each living cell, from bacteria to archaea to man, every little cell is surrounded by a glycocalyx, and this is this gray area here in the electron microscopy when you stain it with heavy metals. But this is not just a unstructured gray dark matter. In contrary, it is a highly organized structure in which we have billions and maybe even more of different structures, and each and every structure has a particular meaning. The sugar, that is so we have a total of 9 to 10 sugars which we are using to build these structures, and we link them together in different linkages, which we call glycosidic linkages, and all this has a meaning. Nothing in this system is without informational content. The sugar of interest in my talk is sialic acid. This is a big sugar, a nona sugar, a sugar with 9 C atoms, and a carboxylate function in position 1. And that makes the sugar negatively charged. And as everybody in the audience probably knows, a cell from outside is negatively charged, and a major contribution is made by sialic acids. And here is made polysialic acid. Polysialic acid is a polymer of sialic acids. And if a molecule is carrying this structure, it has a high content of negative charge, as you can appreciate here by these negative charges in the carboxylate groups. And what happens to a protein or a carrier, I would say, we don't know about other carriers except proteins that can be polysialylated. And this polysialylation then is forming a huge shell around the carrier protein. I should mention the major carrier protein, more than 90% of the polysialic acid in the brain is linked to a molecule which is called NCAM, the neural cell adhesion molecule. And NCAM is a self-adhesion molecule, so it makes homophilic interactions, tight interactions, in the absence of polysialic acid. But if polysialic acid is around, is present as a post-translational modification, these interactions are weaker or maybe even completely lost. And nature invests in order to make polysialic acid. So it has created 2 enzymes, and the enzymes are called polysialotransferases with the given names ST8-CIR2 and ST8-CIR4. It is enough if you remember there is 2 enzymes, the one is called 2 and the other one is called 4. And these make long chains, and these chains are steering, or this post-translational modification is then steering cellular interactions. Here in this, in this scheme, we are looking at the surface of 2 cells. So this is one cell, this is a neighboring cell, and this is in schematic view the NCAM molecule or NCAM another molecule. And NCAM can be polysialylated. And by this polysialylation, the cells are getting, so neighboring cells are getting separated, or let's say the interactions between these cells are getting weakened. In early embryonic life and also in the postnatal life, and we only looked at postnatal stages here in this scheme, almost all the NCAM, this is NCAM, the naked form of NCAM. We have 2 flavors of this protein, and this can be recognized with a specific antibody, and this antibody recognizes NCAM very well if it is naked, but if it is under this polycia shell, it is not very well recognized, as you can appreciate here. So in early phases of life, mouse life in this case, this is postnatal days of specimens, mouse brain specimens looked at, and you can see until day 5, postnatal day 5, 7, we see almost all the NCAM pool underneath a polysialic acid cover. But then after this time point, the polysialic acid decreases rapidly, and we have a steep increase of the naked NCAM. That makes sense in terms of what we saw before. We are soon after birth, so in the early days of our lives, or the early years of our lives, we are declining rapidly in terms of neuronal plasticity, and because polysialic acid is one of the mediators of neuroplasticity, so this steep decline makes sense, and certainly biologically good sense, and we have an increase in the non-polysialylated form of NCAM, maybe to stabilize cellular contacts and make them long-living, because we all know there is not much renewal of neurons in the brain. So most of the neurons are living for many lifelong. So stabilization probably is mediated in part, always in part, by NCAM, the naked form of NCAM. And the same is true for the enzymes. So we have these 2 enzymes. They both come up in early phases of embryonic development and then stay pretty stable over a while and then rapidly decline. And in this case, you can see ST8-CIR, the number 4 enzyme, now it is shown in green, is declining faster than the number 2 enzyme. It's the other way around. The number 2 enzyme is declining faster than the number 4 enzyme. Sorry for that. In order to understand at a genetic level what the contribution of these 2 enzymes is to polysiar production, so we have generated a gallery of mice that are expressing the polysialotransferases in different allelic combinations. So this is by crossing the 2 knockout mice that were made for ST8-CIR2 and ST8-CIR4, and then we crossed those mice in order to get all these different genotypes. And the genotypes I'm going to discuss today, or including in my presentation, are the ones which are shown here. The one with a total ST8-CIR4 knockout is the major genotype that I'm showing, and I have a few slides also on ST8-CIR2. Coming back to the hippocampal formation, so please remember we have these cells which are not polycia positive, that are a pool which is lifelong existing, but and after asymmetric division, we have all these cells polycia positive up until the production of mature granules. And when we look into the brain, what we see is that we have major polycia production in all these hippocampal formation. This is the granule layer, the dentate gyrus. These are these mossy fibers, all highly polycia positive. But what we do not see in histology, and this is a scheme of the histological pictures, is that there is more polysalic acid. Therefore, we have to go much deeper into morphology. But when we do so, what we see is that we have polysalic acid also in this area, and that we have polysalic acid on the very bottom of this granular layer. And when we do hybridization studies, so when we look for the expression of the two enzymes, what we can see is that in this area here, it is mostly the number 4 enzyme that is producing polysalic acid because the expression is higher. But at the very bottom, there is also the number 2 enzyme expressed. It is the same in this area. Always at the very bottom, we do have some ST8CR4 expressed. Now we use the ST8CR4 knockout mice because this is the major enzyme responsible for polycia production in the dentate gyrus. And we were asking if these mice have lost or are impaired with respect to memory formation and learning. And in order to explain to you the task that we used, I explain at least a little bit the term long-term potentiation, because it is a measure for plasticity, and plasticity is a measure for functional brain function or memory and learning. So what you do is you stimulate here in an area, so this is again the dentate gyrus, we have it here, and in this area we stimulate, and then this low stimulation then gives an answer here in this field, which is a recording electrode, and that is on a baseline if you do small stimulations. But if you do a theta burst stimulation, a very strong stimulation for a short period, then the synapses are potentiated and they respond much better to the same low stimulus. And this prevails for quite a while, over hours. It depends on the system where you do your measurements. And when we did this task for learning and memory in the ST8CR4 knockout mice, so the number 4 knockout mice, what we see is that they have lost the capacity in synaptic potentiation, so they are declined. The recording is below the wild type, indicating that we have a decline in synaptic plasticity. And this is, they have a defect in learning and memory. Then there is another test to measure long-term. So this is more a test for short-term. So this long-term potentiation, even so it is long-term potentiation, but it measures more the working memory activity. Long-term consolidation of memory can be measured again in different test systems, and a test system that I'm introducing here is a novel object recognition test. And in this test, a mouse, so this has been a video, but I took it out because it was too long. In this, the mouse is acquainted with 2 objects. It's an open cage, and there is 2 objects in the cage, and normally a mouse goes and explores both objects and spends similar amounts of time with the 2 objects until it is familiar with the objects and then leaves the object. Now you take the mouse out and only replace one of the objects by a new one. And this new object, because it is new for the mouse when it comes in again after a certain period of time, so you leave the mouse out of the cage for a certain period of time, and then bring it in again, and then this mouse starts exploring normally only the new object. But the mice, in our case, what they did is they spent the same time with the new object as they did, after a short period of, after a short interval, so when the mouse was not in contact with the objects, so it immediately recognized there's a new object and only explored this new object. However, if we took it out for 24 hours, the mouse forgot that there is already one object that it knows, and it was spending the same time with the two different objects, indicating that the long-term memory was not working. As I said before, we do have a possibility to compensate for this behavior, and this is mental and physical activity. And this is what we did with the mice. You see, this is a cage, not a naked case with nothing in. This is a cage with lots of toys for the mice, and you see the mouse up here, and it can play all day. It has a lot of stimulation, and it is not as boring as in a regular case. And now, after 24 hours, what you see is the mouse behaves almost the same as a wild-type mouse. So the knockout mouse learns in the same memory task in the long term. Just to give you the situation again as we had it before, the mouse was spending significantly more, so the same time with the two objects, but here we have a full recovery of the memory, or almost full recovery. So the environmental enrichment rescued this memory defects in the ST8 here for knockout mice. Yeah, so what we also saw, and that was really surprising for us, is that when we now looked into the hippocampus of the knockout mice, we saw major polysialic acid production here in this area. The polycia production in this area after learning was even higher, as you can appreciate here on these columns, as it is in the wild-type mice. How can that be? Because the ST8CR4 enzyme, which is knocked out in these mice, can no more produce polysialic acid. So all this polycia was made by ST8CR2, so it was pushed by the environmental enrichment to make polysialic acid in this area and compensate for the memory decline. So we saw all these positive effects, but what we didn't see is when we did a BRDU staining of cells, so when you do this specific staining in the mouse brain, you can watch the survival of newborn neurons over a certain period of time. And this is what we did, and we did not see a full recovery of the survival of newly born neurites even not after enrichment. So there is still an impairment, but we see a massive upregulation of polysialic acid. Even so, it looks like this form of polysialic acid, which is produced by the number 2 enzyme, is not the same as by the number 4 enzyme, and therefore the lifetime of the cells is not fully compensated. So perspectives, this is, I guess, what you wanted to hear today, because this is all, what is the perspective of all these studies? We know from patients, from aging, and also from patients suffering from Alzheimer's disease, that they profit, or this is beneficial if they mentally and physically are trained, mentally and physically. And when we now come back to this picture here and to this new publication by Boldrini, so this paper very nicely shows that we have this process basically going on over all the lifetime. So these cells are preserved. These pools do not decline. So they could produce new neurons, but what we also see is that in these cells where a marker is polysalic acid, the polysalic acid is going steeply down. So these pools are preserved until the 8th decade of age, but polysalic acid is declining, and with the decline of polysalic acid, the plasticity is no more the same. Therefore, stimuli that would increase polysalic acid expression or help reactivate the system that can produce polysalic acid may help to slow down the loss of neural capacity. And the same is true in Alzheimer's. I only concentrate on this one picture here. This is also a very recent publication in Nature Medicine. Again, a very nice one, demonstrating with the stage of the Alzheimer's disease, which is measured in these BRAC stages, so from a low grade to a very high grade. This is a medical doctor who defined this type of staging for the disease. And what you see here is again the polyscia NCAM positive cells. This is the control, and already in an early stage of Alzheimer's disease, we see a massive loss in the polysaccharides production, and this is going on with age, with the increase and the severeness of the disease. So early decline of polyscia positive progenitors also in the Alzheimer's disease. So the perspectives that I bring in my summary slide, we have a lifelong neurogenesis in the hippocampus. The hippocampus is the central structure for learning and memory. Aging Alzheimer's and Alzheimer's disease, and also schizophrenia, I didn't show you those data, but also in schizophrenia, these diseases are characterized by a loss of polysalic acid in neurogenesis in the hippocampus, mostly in the hippocampus. Therapeutic perspectives would be the reactivation of the polysaccharides machinery in whatever mode. This is something we have to further explore. And another option may be the external supplementation of polysalic acid, because up until today, we don't know if it is important that the presence of polysalic acid in a membrane-bound form, or if the presence of soluble free polysalic acid would do the same effect. And studies by Rita Gerardy-Schahn give already pretty good evidence for this possibility, that it is the presence of polysalic acid which is requested, because polyscia may act via ligands in that area and then trigger differentiation programs that then would lead to an improvement of neurogenesis. This is the end of my talk, and before I finish, I wanted to thank my colleagues who really did the work, and this is Herbert Hildebrand in the middle here, and his team, Hauke Tiesler, Melike Kükel-Kerden, Iris Röckle, and Ute Schuster. And these are our funding bodies, and I thank you very much for your attention.
Thank you, Rita, for this enlightening talk. I think we know that we have to take care and boost our sialic acid now by exercise, and physical and mental exercise. The talk is open for questions. And Sami first.
Thank you for your talk, very interesting. Rather to express or overexpress polysialic acid, can you use inhibitors of sialidase?
Well, this is another issue that I did not discuss during my talk. The sialic acids are in threat of being degraded from enzymes that are in the environment. And these are the neuraminases, as the colleague just mentioned. Polysialic acid is not at a high risk for degradation by neuraminases. So there is one single report describing that the new 4 would degrade polysialic acid. We cannot recapitulate those data. And in our hands, at least the survival studies that we have done, polysia is an extremely long-lived molecule. It is not rapidly degraded. So I would say yes and no, as always. Who knows? It may well be that with aging there is a burst of lysosomal enzymes that also impair the polycellulation on the surface and many other structures, or probably cut off NCAM together with the polycellular transferase. That may all well be, but I think boosting the machinery, because you saw that all these differentiating neurons, after the asymmetric division, start immediately getting PSA positive. So in my direction would rather be to find out what really is the function of polysia, because the initial pool is not polysia positive. It only comes after the first division, and then the differentiation process is accompanied by polysia expression. What stimulates this process? And could we make those cells dividing a little bit more frequently to set free the pool for neurogenesis?
Thank you.
Thank you very much, Rita. You show the ubiquity of this complex glycan molecule, and now people are using some sort of analytical, because the analytical devices are being much more progressive. So, for example, in the use of precision medicine, it's possible nowadays to identify the state of glycosylation of IgG and relating that to what people say this is glycan age.
Yeah.
So my question is, since we have that for the analysis of IgG glycosylation, do you think we should think of designing a similar type of test, to have at a very early stage of development, a vision of the situation with respect to our polysialic acid component?
This is a challenging question that you have. I think from a scientific point of view, it would definitely be very interesting to learn about what degrades or what makes this polycellulation machinery getting more fatigue, not being active anymore as it used to be in earlier days, and whether we could find a protein or maybe even IgG in the cerebrospinal fluid that may indicate such a change in the glycosylation machinery. It is very difficult to do this type of studies because samples, whatever I referred on human data, it comes from literature. We never have done any studies in humans because sample acquisition is a major issue. Yeah. But I fully agree, it would be so important to have cerebrospinal fluid, look into these cerebrospinal fluids in detail for the glycomic alterations. I think that would give a lot of information. And yes, probably one should team up with the neurobiologists or with the neurosurgeons to get access to this type of samples. Yeah.
Thank you very much.
Thank you.
Are there some other questions today? Well, if not, we will thank Rita again.